Welcome friends. In this video, I would like to discuss about Operon concept in prokaryotes, that is the gene regulation in prokaryotes. The word Operon literally means to operate. So it is the operation of gene expression or protein synthesis in prokaryotes. Gene is synthesized, the protein is synthesized and the gene is expressed only when it is required by the microorganism. Hence, there is a necessity for the regulation of the gene. So, gene regulation is by Operon. So, Operon concept. Welcome friends. In this video, I would like to discuss about Operon concept. Operon, the word literally means to operate. It is the regulation of gene expression in prokaryotes and their bacteriophages. Gene expression means the protein synthesis, or more specifically, the transcriptional part of the protein synthesis. That means the regulation takes place at the level of transcription. So, operon is a genetic unit. Definition: it is a genetic unit of a cluster of coordinated genes, cluster of coordinated genes in DNA molecule for gene expression. So it is a regulatory genetic unit consisting of cluster of coordinated genes for gene expression at transcriptional level. It is at the transcriptional level. The thus operon thus consists of clusters of genes differentiated to carry on the process of the gene expression. So it consists of clusters of gene on the DNA molecule, and those clusters of genes are the structural genes, the operator and promoter. Structural genes, operator and promoter. Structural genes and operator and a promoter. Now upstream promoter, there is another cluster of genes known as regulatory genes, and this regulatory genes, they produce a messenger RNA to form a protein known as a repressor. A repressor is a protein which is produced by, it is a protein that is produced by the regulatory gene. So the operon consisting of three parts, such as the structural genes, which encode messenger RNA. Structural genes encode messenger RNA Then is the operator, which switches off or on transcription, and the promoter. Promoter bears the sites for recognition, binding and initiation. Recognition site, binding site and an initiation site. For RNA polymerase, to start, RNA polymerase to start or initiate transcription. So the three components of an operon are structural genes which encode messenger RNA, then operator which switches on or off the transcription, and the promoter which provides the recognition site, binding site and initiation site for RNA polymerase so that it can start transcription. Now the regulatory gene regulates the operon by producing a protein which is known as the preparation. So these are the components of the operon, which is regulated by a set of regulatory genes. Now next is the regulation. The regulation is very important because the protein is synthesized only when it is required by the microorganism. It's when the protein has to be synthesized or it is to be stopped is regulated by the set of operon and the regulatory gene. So the regulation is of two types. One is the positive regulation and the other is known as the negative regulation. The regulation can be differentiated into two types, positive regulation and negative regulation. Now, the positive regulation is positive when there is an regulation. Regulation is positive when there is an involvement of an activator in the medium. Activator in the medium, it is not synthesized by the DNA because it is not a protein, and there is an involvement of an activator. Now the second is the negative regulation. In the negative regulation, there is an involvement of, first is the positive and second is the negative. When there is an involvement of a pressure, this type of regulation is known as repressure. This type of regulation is known as the negative regulation. It is with an involvement of a repressure in the negative regulation. There is an involvement of an activator in the medium. This is also in the medium, but the repressure is synthesized. It is a protein and it is synthesized by the regulator gene. It is synthesized by the regulator gene, and the activator is a non-proteinaceous substance which is not synthesized by the gene, and this is away from operon, and this is adjacent to Operon. The regulator gene is adjacent to operon, which produces a repression in the medium. This is adjacent and this is quite away from the operon. Now the positive regulation can be of two types. One is inducive and the other is repressive. One is inducive and the other is repressive. In inducive type of regulation, the gene expression is stopped, it is blocked, it is inhibited, and this is operator, this is promoter, and this is the structural gene. And here it is generally is off, it is blocked, and no synthesis of the protein is taking place. It is off and no synthesis of the protein is taking place. But when there is an activator appears in the medium, that activator is attached to there is an activator which appears in the medium, and this activator then attaches itself to the DNA molecule and it induces the synthesis of the protein or the transcription of the protein, and this is an activator, and the gene expression becomes the gene expression turned on. The RNA polymerase then, due to the presence of the activator in the medium, it attaches itself to the promoter. The RNA polymerase attaches itself to the promoter, and the gene expression is on. It is on due to the presence of an activator in the medium. This activator this is found in Escherichia coli when the Escherichia coli is taking glucose as this is present in E. coli when the E. coli is taking the food as glucose. When there is a presence of glucose in the medium, it activates the formation of CAMP, and that is attached to the CRP, catabolic reactive protein, that acts as an activator, and it induces, this is an activator consisting of CMP and CRP, CAMP and CRP complex induces transcription. It starts transcription. This is known as positive inducive regulation. In repressive, the transcription is turned, it remains on. The transcription remains on because the RNA polymerase is attached to the promoter, and is synthesizing, RNA polymerase is synthesizing protein, and it is on. It remains on. Generally, it remains on. But when the level of the glucose in the Escherichia coli declines, glucose declines in Escherichia coli, that means there is, when the gene is synthesizing protein, it means that it is the activator molecule is attached to this, and it is carrying on gene expression. Gene expression is taking place, but when the level of the glucose declines in the environment, then what happens? This complex, it gets drop off from the site, and there is an inhibitor that inhibits. Here the factor means the decline in the glucose. It acts as an inhibitor, and this inhibitor inhibits the synthesis of, this gets drop off, and no more protein can be synthesized. No protein can be synthesized because this is drop off from the site, and the gene expression is turned off. This type of repressive positive regulation is also found in E. coli. When it is found in the E. coli, when the level of the glucose decline, when declines, the protein synthesis is off. When there is a considerable level of glucose in the medium, considerable more glucose in the medium, then the protein synthesis, it becomes on. This is the positive regulation with the help of an activator. Activator is on the promoter, then the gene expression is on, and when it is not there, the gene expression is off. In the negative type of regulation, the negative type of regulation is also of two types. One is the inducive and the other is the repressive. This is found in lac Operon, when the E. coli is taking. Negative is also of two types. One is the inducive and the other is the repressive. One is inducive and the other is the repressive. Negative repressive and negative inducive. So when this is found in lac Operon, when the medium is having lactose. So when no lactose is there, means instead of glucose, the food here is lactose, and when the lactose is not there in the medium, then it is, the gene expression is stopped. The repressor is there and the repressor is on. The operator, and it stops the gene expression. And It is not possible for the RNA polymerase to move on because the path is not clear, hence it cannot express the gene. So gene expression is generally off, does not take place, and this is when the medium has no considerable lactose. This is found in lac operon. When the E. coli is taking lactose as a food and not glucose. So the repressor is stopping or blocking the gene expression. Now, when the lactose is there in the medium, the lactose is converted into allolactose by an enzyme known as beta-galactosidase. The lactose is converted into allolactose. The allolactose behaves as an inducer in lac operon, and this allolactose, when attaches itself to the repressor, it changes the shape of the repressor, and the repressor can no longer remain there on the operator. It gets off, this complex gets off from here and leaving a path clear for the RNA polymerase to continue, to start and continue protein synthesis. So this is off, this drops off, falls off from the operator, making the path clear for the RNA polymerase to carry on gene expression. This is inducive type. Now this is also found in lac operon. The repressive type is also found in lac operon. The gene expression is turned off. It is turned off. Though the repressor is there, it is inactive. This is inactive repressor, and it cannot block the operator. So the RNA polymerase is having the path free for itself so that it can carry on the protein synthesis. Repressor is inactive and it is unable to, though the repressor is there, synthesized by the regulator gene, but it cannot block gene expression because its repressor is inactive. But when I did a mistake, I am extremely sorry. This is found in tryptophan operon. This is not found in the lac operon. I am extremely sorry. This is found in tryptophan operon. So when there is an appearance of a co-repressor is there, and this co-repressor, when attaches itself, co-repressor attaches itself to the repressor, it makes it brings about a conformational change in the shape of the inactive repressor. The repressor becomes active. It becomes active and attaches itself. It becomes active and it is attaching itself to the repressor and it blocks protein synthesis. The protein synthesis then is blocked. So it is the presence of the co-repressor that repressing, that blocking, that stopping gene expression. So here the RNA polymerase cannot carry on because the path is not clear. This is RNA polymerase and it cannot carry on gene expression. Hence the DNA, hence the Operon is turned off. Hope it is clear to you. Thank you.